So without further ado, let's jump into our scientific talks. Our first speaker today is Dr. Hashim Al-Hashimi, professor at Columbia University. As a grad student at Yale, Al-Hashimi helped develop residual dipolar coupling methodology, and as a postdoc fellow at the Memorial Sloan Kettering Cancer Center, he expanded the domain of applicability of these methods to nucleic acids. As a principal investigator, Al-Hashimi and his trainees discovered many of the ubiquitous motional modes underlying the biological activities of nucleic acids, with important implications for drug discovery and for understanding the mechanisms that cause genome instability in cancer. His group developed methods harnessing the predictive power of RNA dynamic ensembles to identify small molecule inhibitors of HIV-1 replication. In 2009, Al-Hashimi co-founded Nymerum Incorporated to enable RNA-targeted drug discovery using RNA dynamics. His talk today is entitled Visualizing RNA Structural Dynamics Using NMR. Without further ado, Hashim, you can go ahead and share your slides and get started.
Thanks, Kelly, for the introduction. It's a pleasure to be here with you and to talk about some of the work that we have been doing in my lab, exploiting the power of NMR and stable isotopes. And so today, I'm going to tell you a story about how we used NMR to understand the mechanism by which a ubiquitous post-transcriptional RNA modification called M6A impacts the behavior and biological activity of RNAs. So this work was done by two very talented former graduate students. Let me see if I can get my cursor here. Laser pointer. Okay. So Beilu, who's currently a postdoc in Xuan He's lab at the University of Chicago, and Hong Lu, who's currently a postdoc at Jennifer Doudna's lab at UC Berkeley. As Kelly mentioned, I'm a co-founder, just for full disclosure, I'm a co-founder of a company which is now called Base4. And much of this work has been published in these papers shown here. Now, one of the major realizations over the past decade has been that recognizing that there are many more alphabets to RNA nucleotides than the typical 4 bases. So what I'm showing you here is a picture of the various forms of post-transcriptional RNA modifications that have been uncovered. There's actually more than 150 such modifications. And you can see these modifications, which are illustrated in red, can either modify with methyl groups or other groups the bases of nucleotides, or in the case of, for example, 2'-O-methyl, the sugar, and you can also have dual modifications which impact both the base and the sugar. Now, these modifications are increasingly being uncovered not only in certain RNAs like tRNA, but more broadly in messenger RNA and other regulatory RNAs. So one of the major discoveries in the last decade has been that, in fact, these modifications are much more broad and pervasive than previously thought. And of course, the question is, what is nature doing putting these modifications in RNA? Now, I want to focus on one of the most ubiquitous and the most common modification called M6A. And this particular modification is achieved by what are not a family of proteins or enzymes. The ones that install the methyl groups are called the writers. And then those are the methylases. And then there are also demethylases, which remove the methyl group. So this methylation reaction is reversible, it's dynamically regulated, and there are many functions that this methylation serves. Now, for the most part, if we look at the various activities that have been associated with modifications, we find that in most cases, this modification is recognized by specific reader proteins that bind this M6A methyl group, this methylated M6A. But there's also a wide variety of experiments that show that this methylation of adenine doesn't just play the role of being read by proteins, but it also alters the dynamic behavior of RNA. And in particular, it slows down virtually any biochemical reaction you can think of. So if you have a biochemical reaction that acts on your favorite RNA, if you want to slow down that reaction, just put M6A in there, and more often than not, you will observe a slowdown. So for instance, during translation, the accommodation of tRNA during translation is in fact slowed down when you incorporate the methyl group on the messenger RNA. Likewise, splicing to create the mature mRNA is slowed down when you incorporate M6A, and also other processes that involve transcription or replication are also slowed down by the installation of this methyl group. And these slowdowns are on the order of 5 to 15-fold. And the question that I would like to address today is, how is it that this methyl group slows down these biochemical processes to give rise to very important downstream biological effects that I don't have time to go over today? Okay, so what we thought is we came up with a hypothesis, which is that in all the processes that I just showed you, what must happen is that the RNA hybridizes with another RNA. So if you look at, for example, here, when you have the uridine, this tRNA hybridizing with the messenger RNA, there's the process of forming a base pair. Likewise, when you replicate something, you are forming a base pair. Similarly with splicing. So our hypothesis was that M6A somehow slows down the hybridization of nucleic acids. And this was something that we could then understand more deeply with the use of NMR spectroscopy. And in particular, we use a set of techniques that rely on measurements of chemical exchange. These are NMR experiments. They were pioneered by Lewis K. and Art Palmer and others, mostly in application to proteins, and we have been applying them to nucleic acids. And the basic idea behind these experiments is that one can, if you have a ground state and a transient state, so the ground state is 99% populated and some transient state populated to the tunes of 1%, if these two states exchange with each other, then one observes line broadening of the NMR resonances. And from this line broadening, one can perform experiments of the kinds called relaxation dispersion, R1 row, or CEST, and exploit the line broadening to determine the dynamics of the system, to get the kinetic rate constants and the populations and the conformational states underlying the dynamics. And we wanted to apply this methodology, in particular, using what's called R1 row. And I'm not going to go into the details of this experiment, but suffice it to say that we apply a variety of spin locks to modulate the line broadening contributions from chemical exchange. Now, the power of using NMR and stable isotopes in experiments of this kind, we're interested in specific measurements of hybridization kinetics, is that normally when you do hybridization kinetics, you measure things like a fluorophore at the end of a nucleic acid, which might perturb the system, or you do other types of experiments which doesn't give you site resolution. With NMR, we can label individual nucleotides with stable isotopes, shown in yellow, and we can measure the hybridization kinetics from the point of view of individual spins in the molecule and identify intermediates which may be short-lived with the use of these relaxation dispersion type experiments. And so the first thing that we wanted to do was we took this wonderful M6A and labeled the carbon positions shown here in red. This was in collaboration with the Kreutz lab at the University of Innsbruck. And so we took this methylated adenine, we labeled those particular atoms with Kreutz's lab, and then we used chemical synthesis to synthesize our RNAs, shown here, where the adenine is labeled either with the M6A, with the methylated version or the unmethylated version, and then we measure the hybridization kinetics with the use of the NMR experiment. And so here's some example data that we can acquire. So I'm showing you here, and again, in the interest of time, I'm not going to tell you what exactly these data are, but suffice it to say that because you see two peaks, the minor peak tells us that we're able to observe the hybridization of the RNA forming a transient duplex at these high temperatures. And we're able to measure the hybridization kinetics, both in the presence of the methylated form in red on the right and the unmethylated form in left. And you can see that these data are not identical, indicating that the methylation is having an impact on the hybridization kinetics of these two simple strands. And so what we observed was, when we analyzed the data, is in fact, M6A was slowing down the hybridization, the on rate of the two strands by anything between 10 to 20-fold. And this was very interesting to us, because remember, I told you that M6A slows down biochemical processes by roughly a same factor of about 5 to 15-fold. And so our data told us that M6A is already slowing down the hybridization of the two strands, and interestingly and pretty remarkably, had virtually no effect on the rate of melting. So M6A somehow selectively slows down the association of two RNA strands, but it has zero impact on the melting kinetics. And just to contrast this with a mismatch, if you introduce the mismatch rather than M6A, the mismatch would both slow down the on rate, but it would also have a dramatic effect and accelerate the melting rate. And so it was very intriguing to us that M6A had this unique phenotype whereby it only slowed down the on rate, but it did not accelerate the melting rate. And the question was, why is that the case? And the answer to the story, and you can think of this, it can be discovered in how the methyl group rotates. So in the unpaired single strand, the methyl groups adopt predominantly what's called the syn conformation, where the methyl group is shown in the conformation here. And however, when it actually forms a Watson-Crick AU base pair, the methyl group has to rotate 180 degrees from the syn to the anti-conformation. The anti-conformation is less favorable, but required to form the duplex. And so what we thought was probably the slowdown of the association arises from the slowness of the rotation, the fact that you have to rotate the syn to anti, the methylamino group from the syn to the anti-conformation. We hypothesized that's the reason why things are being slowed down. And this is somewhat reminiscent of proline isomerization in proteins, where trans to cis isomerization is slow, and that can in turn slow down biochemical reactions. And so the story that I really want to just quickly uncover today for you is how we went out, how Bei went and Honglu to test this model. So we start off with a syn conformation, single-stranded RNA, and then when we form the duplex product, we have it in the anti-conformation. So we can think of two pathways by which this reaction can proceed. We can first imagine that the methylamino group rotates in the single strand and then hybridizes through what we call a conformational selection type pathway. Or alternatively, you first hybridize in this favored syn conformation to create this unusual mismatched structure where you only have hydrogen bond. And after hybridization, you rotate the methylamino group to produce the product through what we would call the induced fit pathway. Now, the take-home message from my talk today is that we were able to, using NMR, measure every single rate constant associated with every one of these branches. So that's a total of 8 rate constants through the power of NMR and stable isotopes. And from these experiments, what we discovered is that the reaction roughly proceeds 50% through the induced fit pathway and 50% through the conformational selection pathway. So we measured all of these rate constants. So let me just show you some examples of these measurements. So here we can take the single strand and simply measure the NMR data on this single-stranded RNA, focusing on the carbon probes I showed earlier. And from this type of CEST experiment, this one here is shown for the methyl group, we can directly measure that we have in the single-stranded RNA, 91% of the RNA is in the syn conformation, 9% is in the anti-conformation, and the kinetic rates of interconversion, information that is very hard to get and seemingly impossible to get, I would say, with any method but NMR, because we're literally measuring rotation of a methylamino group with surgical precision. Those rate constants are provided over here. Now, similarly, we can take the product of the reaction, starting from the duplex state, and measure the kinetics of forming this unusual singly hydrogen bonded state. And this is shown here in this particular experiment, where we now have a well-formed duplex, which is locked into this duplex state by virtue of having this tetra loop. Now, when we make the NMR observations, what we can measure is the fact that the methylamino group, even when it's base paired, can rotate back and forth. And we can measure that it's 1% rotated in the syn conformation with these rate constants shown over here. Okay, so using these approaches, we were able to determine every rate constant that I show here. And I want to show you that our model is quantitative and predictive, and that using these rate constants, we can quantitatively predict how M6A actually slows down the hybridization kinetics. And this is just the correlations to show that. But I want to show you in the end how it is that this particular isomerization only slows down the on rate, but doesn't impact the off rate. And here's the reason why. So the on rate through the conformational selection pathway, let's start there. The reason things are slow is this intermediate has a very low population. And so because of the low population of this intermediate at 0.5%, the hybridization is slowed down in the presence of M6A. And as a consequence, the on rate is slow. However, when you go the other way around, once you are in this conformation, M6A is stable, and so the dissociation, the melting rate is not impacted, because once you start from this conformation, M6A behaves just like an AU base pair. So the melting kinetics is not affected. From the induced fit pathway, what we have is, again, that we starting from this conformation, we can form this mismatched structure, And while this dissociation is fast, the population of this intermediate is low. And as a consequence, you don't accelerate the melting rate. However, you slow down the forward rate even from the induced fit pathway because of the slowness of this step here, which is the association of M6A when it is in the syn conformation. So with the use of NMR, we were able to understand why it is that M6A only slows down the on rate, but doesn't increase the backward rate. And we could test this model on an RNA molecule, which we study in the lab, which interconverts between two different conformations. And we could use our model to predict how M6A will slow down the dynamics. And in this particular case, because the kinetics are inherently faster, the slowdown effect is even greater, and it's about 300-fold. And this could be quantitatively predicted using our model that I just described. So in conclusion, M6A has the ability to slow down RNA processes and not selectively by slowing down the hybridization of nucleic acids. So anytime you think of a process that requires RNA hybridization, then one can predict that M6A will slow down that reaction. And depending on the speed of that reaction, the slowdown can be 10-fold or 1,000-fold. And for CIL, you have your work cut out, because there are all these wonderful modifications, all of which would be interesting to study using NMR, and it would be great to have all of them isotopically labeled to enable the kinds of studies that I just described. And so I'll finish by acknowledging the work done by the students, and thank you for your attention.
Wonderful. Thank you so much, Hashim. That was a great talk. We do have one question that came in through the Q&A right now. Are you only monitoring the CH3 resonance in your experiments?
Yeah. Not only the methyl groups, we're monitoring the methyl groups as well as the C2 carbon on the base and the C8 carbon on the base. And that's actually quite important because different nuclei are sensitive to different processes. So for instance, the methylamino rotation is detected by using the methyl group, whereas the C2 and the C8 are used to detect the hybridization step. And so by having multiple nuclei, we're able to dissect these two distinct processes and resolve them in the NMR experiments. And if we only had the methyl group, it would be hard to disentangle the contribution of the isomerization from the hybridization. That's a good question.
Awesome, thank you. What amount of material did you use and produce? Is another question that just came in.
Yeah, these samples are, your typical 0.5 millimolar RNA samples, NMR samples, and so so many mgs, maybe a few mgs per mil, and so that's the type of it's no different than any standard NMR experiment, but the more the better, and the cheaper the isotopes, the better.
Thank you, Hashim. Another question we have: how rare are these modifications that you mentioned in nature?
Yeah, great question. So M6A is the most abundant, and it's at the tunes of 0.1 to 1%. And so, but some of them are less than that. And so they're rare. So roughly every messenger RNA is modified at least once with M6A somewhere. Mm-hmm. And this is, again, a nice example. In NMR, we always appreciate rare conformational states. And here's an example of rare modifications. Their abundances are actually not that high, but they have radically important roles. If you knock out the methylase, as Swanee showed and others, it's lethal. So these rare modifications are fundamentally important for biology. So they're rare, but they're quite important. And there's still studies being done about figuring out exactly the stoichiometry. So, once you look at a given site, you have, in a messenger RNA, you have one site that will be methylated, but it could vary. One messenger RNA might be this site, in another messenger RNA might be on a different site. Getting that stoichiometry is actually quite difficult, and people are working on it. But on average, is about 0.1%.
Awesome, thank you. And I'll throw in one more question of my own. So you mentioned we have all of these rare modifications. So outside of M6A, what other, I guess your other top two would be of most biologically relevant insights for the greater community, in your opinion?
Yeah, so 2'-O-methyl is a great modification because of interest, because first of all, it modifies all nucleotides. It modifies the sugars and affects the sugar pucker properties. And another former postdoc, Halabasi, did a wonderful study showing how, in a way, NM, this 2'-O-methyl modification, is the inverse of M6A. It slows down the melting. but doesn't have an effect on the association. So it's kind of like the yin and the yang. And the interesting thing is there's a dual modification, which I also like, which combines M6A with NM. And so then the great question is, one of them slows down the forward rate, the other one slows down the backward rate. What happens when you put both? And the answer is you end up slowing both. And that's paper's work that we're doing. Inosine is another modification that is also of great, and pseudouridine. So pseudouridine, inosine, 2'-O-methyl, M6A, and the dual modifications, because you can have the combinations of these, would be my top choice.
Awesome, thank you. And we did have one more question come in. Unfortunately, we don't have time to answer it right now, but I can send it over to you and we can address it after the fact. So definitely lots of experiments needed for NMR to get the answers to these questions. Thank you, Hashim.
Thank you.